So what is this VCF format? So for those of you who are going to do variant calling, you will have to know anyway, because the results of the variant calling will also be VCF files. So known mutations are stored in VCF files, but also your mutations, the one that are called based on your data, will be in a VCF file. So VCF files are again text files, as always, with a specific format. So you have the red part that describes the variant. So what typical information do you get here? Well, where it is, so the position and the chromosome. This column will tell you whether it's a known variant or not. This column will tell you what the base in the reference is, and this column will tell you what the base in your reads is, or what the, when it's known variants, what the alternative is. You will get the quality score for the variant, and here you will get information on the calling, the variant calling that was done by GATK. So GATK is going to use a lot of filters to call variants. He's going to take a lot of things into account. So if a variant has passed all the filters, you get pass. If it hasn't passed one of the filters, you get the name of the filter here that was not passed. So this is a low quality one. So information on the variant, and then typically you also get information on the sample. So the BAM file that gave rise to this call. So what can you find there? Well, the format explains here, this explains what you see here. So this is the explanation basically, and this is the actual data. So GT, the first thing that you see here is the genotype. So you have either heterozygous or homozygous variants. That's what this thing is saying. And then you get here the second thing, the AD is the allele depth. So how many reads show the variant? That's what you see here. These second numbers. So here, for instance, 173 do not show the variant and 141 do show the variant. That's how you can interpret these numbers. DP is the depth. What is the depth? How many reads map to this position? So that's the third number here. So you always have these columns that distinguish different values. So the third number here. So 282 reads in total mapped to this position. That's what you can see here. And then the last numbers that you see here, these things, they are, well, quality scores for the allele. So 255, as always, is very high quality. 0 means low quality, quality scores. So basically, this tells you more about how this variant was called. Where does it come from? Is it heterozygous, homozygous? How many reads were there? Can I trust it? So if there are a lot of reads, you trust it. If there are not a lot of reads, well, it's not so trustworthy. Okay, so this is how you can interpret these VCF files. So they're available for known variants, and GATK will also generate them for your data.